Welcome to TED Talks. You're still with me, Simon, and in this session, we are going to focus on the third functional structure of proteins, which we are calling the tertiary structure of proteins. We had earlier already said that proteins take up structures. The first structure was the primary structure, that's according to the sequence of amino acids. The second one, it was now the packaging, which was done by hydrogen bonds in addition to peptide bonds. The tertiary structure, we can now say this protein structure is further packaged with more interactions on top of hydrogen bonds and peptide bonds to give it a three-dimensional structure. Yes. So more bonds are coming in. These bonds include one ionic interactions. In the ionic interactions, remember that in our amino acid that are now making up, when dissolved in water, we can find when we have a negative pole in the zeta ion, which we talked about earlier. Then we can also find when we have the positive pole on another amino acid altogether, either in the same chain or different chains. So an ionic interaction will link up this. So this is what we shall now call ionic bond. So various chains can link up together via ionic interactions. That's number one. Number two, we can also continue to have hydrophobic interactions. We talked about what the meaning of hydrophobicity means. In this one, there are some amino acids whose R groups are purely hydrophobic, and if there is an adjacent amino acid also whose R group is hydrophobic in another polypeptide chain, it will mean that these can interact these can interact. Now that one will be a hydrophobic interaction. Hydrophobic interaction. Can continue further and see another bond. Can talk about the third bond, which we can call now the disulfide bridges. The disulfide bridges. The disulfide bridges. The disulfide bridges, remember we have some amino acids like amino acid cysteine and methionine. These, their R group contains some sulfur. In our definition of amino acids, we had expanded it. So these contain sulfur there. So let's say if methionine containing sulfur here and cysteine containing the sulfur there, a special bond will be created in between the sulfur atoms, which we are calling the disulfide bond, or we can call it a bridge, because bond is bridge. So those are the additional bonds. That means on top of hydrogen bonds that can happen either intramolecular bond hydrogen bonds or intermolecular hydrogen bonds, and on top of peptides that maintain the secondary structure, ionic interactions come in to maintain the tertiary structure. Hydrophobic interactions come in to do that, and also disulfide bridges come in to do that. So the tertiary structure, tertiary proteins majorly constitute the globular proteins, which we shall expand in our subsequent discussions. Globular proteins. The tertiary structure gives us fibrous proteins. So it means we shall expand them at their time. So that is the tertiary structure of a protein, which gives it a specific functions. The more the bonds that are within, the more it is packaged to suit its function.